let us start with dihybrid cross, wherein in dihybrid cross we will take two contrasting features, that is the seed shape and the color of the seed. Parents, that is the round yellow, that is capital R, capital Y, into wrinkled green, that is small r, small y, small y. We take each of the gametes, that is capital R, capital Y, small y, that is round yellow. Selfing, that is the capital R, capital Y, small y. Now let us do the crossing and we obtain various varieties. On observing the F2, Mendel found that yellow and green color segregated in a 3:1 ratio. Round and wrinkled seed shape also segregated in a 3:1 ratio. Dihybrid phenotypic ratio, round and yellow 9, that is round green, that is 3, wrinkled yellow 3, wrinkled green 1. It is derived as a combination series of 3 yellows, 1 green with 3 round, 1 wrinkle, that is 3:1, 3:1, that is 9:3:3:1. Dihybrid genotypic ratio, capital R, capital S, capital Y, capital, that is 1, R, capital Y, small y, that is 2, capital R, small r, y, that is 2. Therefore we get the ratio 1:2:1:2:4:2:1:2:1. Okay, now we come to the non-Mendelian inheritance, that is first one is the incomplete dominance. Incomplete dominance, it is the inheritance in which the offspring shows characteristics intermediate with two extremes parental characters. Example, flower color in snapdragon dog flower or anirium. Example, flower in snapdragon dog flower or antirrhinum species and mirabilis jalapa 4 o'clock plant. Now we take the gametes and when we do the cross we get the R, capital R, small r, pink color, that is half capital R, half small r. Now we take the Punnett square, that is the egg and the sperms, and when we do the recrossing we get 4 types of ratios. We get the varieties, that is genotypic ratios 1:2:1, 1 red, 2 pink, 1 white. Genotypic ratio 1:2:1, 1 RR, 2 capital R, small r, 1 small r, small r. Here phenotypic and genotypic ratios are same, that means that R was not completely dominant over small r. P plants also show incomplete dominance in other traits. Next we go to the codominance, that is, let us see the blood group. The A blood group, we have the gene IA or IA-I0. B, that is IB or IB-I0. AB, that is IA-IB. O, that is I0. Now let us take an example. If the mother has A blood group and the father has B blood group, what is the possible blood group that we can see in the offspring? Mother, that is IA, IA-I0. MB, father, that is IA-IB, that is IB-I0. We have already seen that the dominance is 100%, incomplete dominance is 50%. It is inherited in which both alleles of a gene are expressed in a hybrid. Example, ABO blood grouping in human. ABO blood group are controlled by the gene I. It controls the production of sugar polymers antigens that protrude from plasma membrane of RBC. The gene I has 3 alleles, IA, IB and I0. IA, IB produce a slightly different form of the sugar. Allele doesn't produce any sugar. Here is the complete table where we can see that we find such type of genes and what will be the genotype and phenotypes. When IA and IB are present together, they are both expressed as their own types of sugar. This is due to codominance.